Great. Well, very good to be talking with you all this morning. If you missed me earlier, I'm Frank Nothaft, and this is my colleague Sriharsha, and we're part of the Genomics team here at Databricks. So we'll be talking to you about some of what we've done in our genomics product, focused on both our pipeline and tertiary solutions. And in case we didn't motivate it this morning, genomics is very big data, and here's kind of our up and to the right curve that we in computing love. This shows you the number of genomes that we've sequenced per year, and this comes from a paper, kind of comparing genomics data to astronomical data, showing that genomics is really the new big data scientific field. And when you look at what people are doing right now, obviously we heard Regeneron talk about their massive scale population sequencing studies, but these population scale sequencing studies are no longer particularly rare. There are a number of different pharmaceutical orgs that are doing this population scale sequencing. There are a number of different health systems that have a specific geographical area or a specific kind of patient population that they cover who are doing this large scale population sequencing. And it may be, if you're in a pharmaceutical org, you may be doing it to try to accelerate drug development. If you're doing it in a clinical org, you may be trying to go ahead and use it to guide, get a better risk profile for the patients who come into your system. And for those of you who aren't familiar with it, we kind of breezed through what genomics, or what your genome is earlier. But when you look at the human genome, you wind up having 22 autosomal chromosomes. So these are the chromosomes that you get one copy each from your mother and your father, and then we have two sex chromosomes. So if you're a woman, you typically would have gotten an X from both your mother and your father. If you're a man, typically X from your mother and Y from your father. And this is what it would look like if we looked at these karyotypically. So if we were able to just take every single chromosome out, and like we're doing a karyotype, which many of you may have seen before. Practically speaking, though, of these 23 chromosome pairs, we wind up having about 3.2 billion bases of sequence. So each base in this chromosome, each individual letter, we can think of that as an ACT or a G. We'll have 3.1 billion of these. Our longest chromosome is chromosome 1, which is about 250 million bases long, and our shortest is chromosome 21, which winds up being about 46 million bases long. Of this, 1% of the genome encodes proteins. And this is kind of interesting, right? So Jeff talked earlier about how Regeneron does exome sequencing. So that 1% of the genome that actually encodes proteins, that is what we call the exome. Beyond that 1%, though, there's actually 80% of the genome that is functional in some way, shape, or form. And this number comes from the ENCODE project, which looked at areas of the genome that are responsible for some sort of a functional or a regulatory process. So although we just have 1% of the genome where a change in the letter that we see can impact the actual protein that is coded, we have this 80% of the genome where a change in the letters that we have can impact whether a protein binds the genome and initiates transcription of the gene. So we do want to do these very large scale studies. And if you're not familiar with how you go ahead and analyze a genome, when we start with genome sequencing, we have this. Typically we think of it as we take our genome, we fragment it up into a bunch of pieces, and then our sequencer reads these. So this is an analogy that I got from Mike Schatz, who's a professor at Johns Hopkins, who does a lot of work in genome assembly. And I think it's a really nice, clear way to look at it, because you don't have to know anything about genomics to understand this. So here, for example, we have the best of all times, it was the worst of times. When we do sequencing, what we do is we take this long stream of genomic letters, we break it up into a bunch of small fragments, because our sequencer can't read the genome end to end in one shot. We break it up into a bunch of small fragments. We get a pool of these kind of little strings back. From that, we'll take these little strings, we'll align them back to the genome. So, when we think about the genome, we had this human genome project that completed in 2001. We say we understood the genome, the human genome. What that really meant was that we had a sequence that corresponded to what the average sequence for a genome in a single organism looked like. What we can do with this is that we can take that bag of reads that we had earlier, we can align it back to the genome reference. So we'll say, okay, it was that aligns back to the first base, times it was that aligns in the middle. That allows us to kind of get this global ordering and this global place where every single fragment that we sequence map to. And then finally, we'll go ahead and take those reads that we've aligned back, and we'll use that to reassemble the original sequence. And here we didn't have any variants. There's just not that many interesting changes that I can make to a timeless classic. But in our genome, in a single individual, we would typically expect to see a change about 1 in every 1,000 positions. So that's kind of the high bit for how we analyze it. And this is just talking about how we would do variant detection. But there are a lot of different assays that build on top of this general primitive. We can take this and use it to discover genomic variation, but we can also use this to find areas of the genome where we are open for binding proteins to the genome. We can use this to identify genes that are expressed more commonly, so genes that we would expect to have a higher concentration of proteins actually made. We can use this general primitive to do a lot of disparate genomic analyses. And ultimately, though, this does get to a pretty large scale problem. And if we start by looking at a single human genome at 30x coverage, this is if I sequence every single base in the whole genome, we'd be looking at about 100 gigabytes of raw data. And this wind up, if I look at this winds up being about 1.5 billion reads. So if I look at this as a database, I have this database for a single human genome where the raw data is about 1.5 billion rows long. If I go ahead and I scale this out to 10,000 samples, which is maybe the point where I might start to see some interesting population level effects, then I'm looking at 1 petabyte of raw data just from the genome sequences themselves. Ultimately, like a lot of the interesting experiments that we run operate, we'll process the raw reads into a form of data that we can then operate on. For a lot of the interesting experiments, we're often operating on genotype data, like what you heard from Jeff and what you saw in my demo that was working on this variation or genotype data. Even then, that still gets very large once we've aggregated a lot of samples together. So if I look at a GVCF, which is kind of the summarized likelihoods, it's a statistical observation at every single site in the genome. We'll easily be winding up with 10 terabytes of data that I'll be running an analysis on for my 10,000 whole genomes with over a trillion rows. When I get to VCF, which is the kind of the final, you could think of it as a matrix that has all of my variants and all of my samples, plus all of my samples, and kind of it lists out which variants are present in which samples. For 100,000 whole exome samples, this can easily wind up being 40 terabytes. So it's still a pretty large matrix or pretty large data set to be running a bunch of queries on. And additionally, with this data, we often wind up having to run some sort of a join or some sort of a query. Oftentimes, these wind up being based on the genomic plane. I might want to take all of my lists of all of my genes. I might want to join that against my data set, use that to select only the reads that map to genes that I was interested in, or only the variants that showed up in genes that I have, that I know associate. I have some external annotation that tells me these variants in these genes have been seen in a disease before. I often have to run that join to go ahead and find those data points that I'm then going to go ahead and analyze. And again, I have to run this. It's a very high cardinality join. So ultimately, when we if you look at genomics right now, this is typically based very heavily off of tools that are kind of designed in a traditional high performance computing architecture. And they're they're typically designed along these very monolithic tools. So we look at a genome sequencing pipeline. If you look at the GATK, which is one of these common tools for analyzing genomic variation, if you look at all of the steps involved in running the GATK end to end, you wind up having about 20 different processing steps. And if you follow their best practice recommendations, you can very easily have about 10 different tools coming into play just to build that pipeline. Additionally, these tools are all designed to run on a single node. Some of them are even actually designed strictly to be single threading. An interesting thing in the GATK4, which is the newest release of it, is it completely eliminated the multi-threading support. So now if you want to run the GATK not on a single thread, you actually have to manually parallelize it yourself or use Spark. But even then, let's say I have gone ahead and run these tools. I have a tool that is multi-threaded. They're still typically not optimized for the cloud. They don't really make any sort of approaches that make it very easy to work with data that is sitting in a cloud store. If I want to work on data that I'm going to read from and write back to a cloud store, I have to have some process that marshals that data out from the cloud store to my local node, runs the tool, and then stages the data back. And ultimately, this makes it very, this makes this analysis process very rigid. It's hard to collaborate because you have these long, complex dependencies to share. You often wind up having incomplete data management tools. So it's just very hard to build and share these analyses. So I'll turn it over to Ram now, who'll talk you through what we're going to do.
Can you hear me? Cool. So we kind of started off here about a year back with a lot of our customers. A lot of them actually wanted to use Spark to scale the single node tools that they had. We worked very closely with them, and we found a few patterns. It was not easy to take these single node tools and scale them up. Even things like GATK, haplotype caller, and so on that are supposed to work on Spark are not easy to scale and are not concordant with the single node versions. So after looking at all this, we decided that we need to build a product here. And this product has to have a few goals. So there's a few things that we need to kind of achieve. The first thing is we actually wanted to make genomic workflows easy to set up, run, and reproduce. So many of these workflows are very well understood. The challenge is just scale and cost. So we wanted to, and of course, making it simple. So those are the three things we wanted to solve: simplicity, scale, and cost for the genomic pipelines. We also wanted to be able to scale in 2 dimensions, both in the number of samples that we process, as well as in the total number of gigabases. So how many bases we actually read. Sorry. We wanted to support common formats here. So there's a lot of legacy formats that people use, VCF, BAM, and so on. Many of them are actually not efficiently splittable for the cloud. So when you're reading it from cloud storage, for example, it's not easy to read them in a parallel fashion. But we still wanted to support them because data transformations themselves are painful here. And as much as you can avoid having to transform data from one to the other, the better. We didn't want to reinvent the wheel. There's a lot of best-of-breed tools available here, like Atom, GATK, and Hail. And we actually wanted to support and extend them as opposed to building something new. We also wanted to be language agnostic. We have customers who use SQL, customers who use Python, R, and so on. And we want to have first-class support for all of that. We actually wanted to build optimized tertiary analysis primitives. So there's a lot of common genomic patterns that we were seeing that could actually be optimized in the runtime itself and made fast and efficient and simple. And we wanted to do that. Last but not the least at all, we wanted to provide documentation and examples that people can just start off with so that it's easy for you to hit the ground running here. So these were our main goals. We are very excited to announce that we are in preview now. We have a unified analytics platform for genomics, which we talked about in the keynote a bit. This is in preview mode. We've already been working with a few customers on this. So if you have interest in this area, please reach out to us. There is also an HLS event today evening that we are going to be at. So please find us there. So to dig in a bit more, what can we actually do today? So we do have a one-click setup for DNA sequencing workflow. So this runs through alignment, variant calling, and annotation on, let's say, a 30x whole genome sequence in under an hour and a half, and it's very cost efficient. We also have, the pipeline is also incremental, so you can actually process new samples as they arrive. You can also process a large batch of samples if you want. This is not easy to do, actually, and the fact that it's all automated and simple means you just point at some new samples and you can run the pipeline. Right? On a large class of common genomic queries, and I'll hopefully be able to demonstrate a class of queries to you, we do have a 100x speedup versus other open source tools in this space. We also provide pre-canned notebooks for a lot of canonical genomics workflows. So it's easy for you to get started. We also host a few sample data sets so that you're not going off looking for data sets to try these notebooks on and so on. So all this we are doing to kind of get you up to speed on this as fast as possible on our platform. So the main goal in simplifying the workflow is there is broadly 2 classes of things that you might want to do. There's tertiary analysis, where you have genomics notebooks that we provide, or you have your own notebooks that you create. You might even be using SQL, or you might be using open source tools like Atom. All of these run in using the same optimized engine that we built. That runs on a tertiary platform that we provide. The tertiary platform is basically Databricks clusters with an optimized image. This image is optimized for both genomics tertiary analysis. It also contains the reference data, so you're not looking for genome references. They're indexed properly. It also contains sample data sets, so you can actually quickly use them and play around with them and figure out what analysis you want to actually describe. And when you're running this image, you're basically accessing your data using your secure access. So that lies in your control plane. You can point at aligned data, you can point at variants, you can point at GVCFs, you can point at annotated data and run complex analysis. If you don't have aligned data, if you don't have variant calling data, but you just have raw reads, we also have a DNA sequencing pipeline. This runs as the job. It uses the same genomics image because there's a lot of common primitives between tertiary analysis and the pipeline. The pipeline can take any raw reads, whether it's in FASTQ format, BAM, and so on, and convert them into an optimized internal Parquet format that is, again, stored back in your account. So basically, it's data that's processed through this pipeline. We align the raw reads, we call variants on them, and we annotate them with single nucleotide polymorphism annotations, SNPF annotations. So the goals of the pipeline and the tertiary analysis are slightly different. Tertiary analysis is optimized for low latency and to be able for you to interact with your data as fast as possible. The pipeline is optimized for cost, reproducibility, and simplicity. That's where we are at in terms of simplifying the workflow. Now, apart from simplification, the other thing that's important to us was scaling. We wanted to make these things operate, these pipelines as well as tertiary analysis operate at a very large scale. We scale in a couple of ways here. There are stages in our pipeline, for example, that use BWA for alignment. There is a stage that uses SNPF annotation for annotating your variant calls. Both of these are actually parallelized using Atom pipes. There is also an implementation of the haplotype caller that we have. This implementation is more scalable than the GATK open source implementation that you might have seen. It is also concordant with the single node implementation of GATK, which is actually not true of the open source caller. So we achieve higher concordance with higher scalability, and that's actually hard to do. Where possible, we also allow you to downsample regions of high coverage. And if needed, you can call variants only on the target regions that matter to you. All of this is flexible and configurable. So it's easy for you to achieve scale by doing all of this. While achieving scale, we also wanted to keep costs low or try to make this pipeline as cheap as possible. And how do you do that? So when the nice thing about being cloud native is you can actually grab as many resources as you want. But the hard part of being cloud native is making sure that your scheduler and the Spark runtime and so on knows when to release these executors, right? Within Databricks, we have auto-scaling features that are very clever about how they grab on new resources and how they release them. And the Genomics pipeline actually uses this a lot. So for example, we scale up on demand. If there are stages within this pipeline that actually can parallelize really well, we scale up. When there are stages that have a lot of skew and they don't quite run as parallelly as they can, then we can aggressively scale down. And here is an example run of a job where we can demonstrate this. So for example, here, the first part of what I'm showing you is, so there's basically three things here. There is the active executors, there is the total number of executors, and the executors holding on to shuffle data. In an ideal world, the executors holding on to shuffle data and the total number of executors that are running in a Spark job should kind of be the same. And that's what you kind of see here. We are almost running ideally, right? At the beginning, there is a stage of alignment, which is parallel and fast. We finished that. So the cluster starts off with a small number of nodes. It auto-scales up. Now, if you're in open source Spark at this point, it auto-scales up and you'll just stay up. So you're never going to let go of those resources. What happens inside Databricks is, at the stage where we reach variant calling, you notice that there is some tasks that take a long time, and there is skew in that stage. So as there is skew, the rest of the tasks complete, and we let go of those resources. And that's why you see the curve dipping to the bottom. And there is a very small stage at the very end, which is annotation. Annotation is not a computationally very intensive stage. So we grab onto a few more resources as we need, quickly finish the stage, and shut down. So the whole process now means I'm saving about 40% in cost by just using auto-scaling in Databricks, right? So these are the techniques that we use to actually keep the costs manageable and small, as small as we can. The last part is scaling in terms of how we can analyze, how we can run tertiary analysis. So this is when you have the secondary analysis done, either through our pipeline or through some other pipeline. You have these aligned reads, you have variant called data sets, and so on, and you want to now analyze them. So the kind of things we do here to scale this step is when we are ingesting the data, we can actually optimize the data layout because we understand genomic data, right? Common things that you might want to do is partitioning by sample. You might want to sort within a partition by certain things that put, for example, reads that are near to each other actually physically near, right? We also prefer nested Parquet formats to white tables, and we can do this in a way that doesn't affect query times. So for example, by having nested Parquet formats, it's easy for the optimizer to look at one column as opposed to 500 columns. But because in Databricks, we can actually read from nested data structures as fast as you could from a white table, so you're not compromising on anything here. We leverage Parquet metadata very effectively. So there's a lot of metadata that once you have the right type of statistics, you can use this metadata to skip row groups that you're not interested in. And we do all of this automatically for you. We leverage delta extensively for data skipping. So when we are optimizing this way this genomic data is stored, we create a lot of statistics for you that allows you to only look at the regions of interest and not have to sort through all the data, sift through all the data. We leverage DBIO for caching, and we also use, like I mentioned, fast decoding of nested structures. And where needed, we extend Catalyst with optimization specific to genomic queries. So there are optimizations that you could do that wouldn't quite work on generic data, but actually works on genomics data because you understand the data layout and you understand the query patterns. So we do that. And in cases where there is very expensive operators, we figure out how to code generate it so it kind of runs pedal to metal. Right? With all of this, we are able to scale tertiary analysis. This is combined. This is where you can see like 100x improvement in performance. So that's kind of what I have, what we have as kind of the material. What I wanted to switch through was, since I have about 6 minutes, I wanted to quickly give a demo and maybe take questions at the end. So is this clear to everyone in the back? Cool. So what I'm going to do here is I actually have a notebook that illustrates a very common pattern of query. So the pattern of query here is basically, forget the imports, so this is just some stuff that we do to inject some optimizations. What I'm going to look at is, if you had been in our morning keynote, we were actually examining this data. So this is data from like Platinum Genomes dataset, which is a very high coverage dataset for the very high coverage alignment as well as a variant called datasets. I'm going to look at a single sample here. So this is roughly, as Frank mentioned, about 1.5 billion rows. So we're actually looking at a data set that has about 1.5 billion rows here, right? The data has a lot of things in it. It has a very complex schema, but basically the things that matter to us are the start position, the end position, what is this read about, what is the sequence, and so on. So these are short reads. What I'm going to do is I'm going to look at these reads and look at targets and figure out what are the reads that actually overlap with the targets that I'm interested in, right? So there may be some targets that I'm interested in. I want to know how much read support do I have? So for targets, we have a data set that we've already provided here for the demo. This is, again, these are public data sets that you can get access to. Now, if you look at this data set, for example, again, it has a start and end position. There is a gene ID, like what gene does it correspond to, and so on. Now, the problem that I'm trying to solve is I have a large number of reads who have their start and end positions. I have a small number of targets that have start and end positions, and they also have chromosome information. So I want to look at the reads that belong to the same chromosome as the targets that I'm interested in, and I want to kind of line them up where these two intervals overlap. So if you're familiar with this as SQL, I'm looking at an overlap join kind of problem, right? So the basic question is just this. Now let's look at how many of these have support in a particular region. So this is actually looking at all the regions. So for example, we are looking at all the reads and looking at all the targets and doing a join to figure out what are all the reads that have support in any of the targets, right? This is if you think about it, there is about 900,000 targets. There is about 1.5 billion reads. This is a join that's pretty massive. And we saw that this finishes in about 17 seconds. I could filter down to a gene that's interesting to me. This is like a GIPC2 gene. And I can say, is there any read support, or how much read support do I really have on this gene, right? Maybe my data was not even targeting that. So I just want to look at that information, right? Once you've done that, you can look at the variants for the same sample, same person, and figure out how many variants are actually targeting this particular gene. This is yet another overlap kind of join. It's a very similar pattern, except now you're looking at variants and you're looking at targets, right? So you can see that it took a pretty reasonable amount of time. We're talking about joining 1.5 billion rows with another 900,000 rows, right? And it was pretty reasonable. Now I have another query that is actually open source, that's running on a stock open source join. So this is, you could actually play around with this yourself if you wanted. All I've done here is I've actually loaded data using Adam, and using Adam's broadcast region join, which morally does the exact same thing, right? It looks at targets and it looks at reads and looks at where there are overlaps and does the join. I kicked this off 30 minutes before I came here. So it's been about 50 minutes, it's still running, right? So that is the difference. So you can see that the type of optimizations we talked about provide literally 100x improvement in performance. Okay? So I think that's all I had. That's all we had. We'd be very happy to take questions. I think there's about 2 minutes or so left.
Thank you. We got a couple of minutes for questions, and can a volunteer actually Tony, can you pick up a mic from there, see if the audience on the other side wants to ask any questions? Any questions? Okay.
Hi. Thank you. You mentioned aggressively scale down when you do the scale up, scale down on the cluster. What parameters do you guys use to do the scale down within the state? Is it an Amazon parameter or your own defined?
Yeah. So we don't really do anything genomic specific to leverage this auto scaling. So if you were to run an auto scaling job in Databricks today, that already does this. It is very effective for us, right? We don't really tweak any parameters in auto scaling. With the way it works is, the cluster starts off with a small number of nodes, but when there is a large number of tasks and they're taking long, the auto scaling algorithm knows that okay, I can request for more nodes, right? I want tasks to make progress. So there's a lot of backup of tasks, and there's only a few nodes, and you've set the max number of workers to be, let's say, 30. It starts with the min number of workers, let's say 2. So it's going to launch 2 nodes, it's going to run some tasks, and they're taking long, but you've allowed it to launch up to 30 nodes, so it's going to start launching, right? So that part is the easy part. Like scaling up is the easy part. Scaling down is the hard part, because when do you know when to scale down, right? And one way to know when to scale down is to say, when I'm running a stage where some task on the stage doesn't require shuffle data from any other tasks, outputs, then those executors can be killed. So that's the right way to kind of figure out when this can be scaled down, right? That's the difference between how this algorithm works, for example, in Databricks versus elsewhere, which is scaling up is the easy part, the scaling down is the hard thing. What I showed you there was it scales down even within a single stage, right? And it also scales down across stages.
We got a last question over here. Only got 3 seconds left, so let's make it short.
Hi, nice talk. So for the demo, how many nodes was your cluster using?
8 nodes.
8 nodes?
8 nodes, yeah.
All right.
Okay, that's all the time we have. Both Frank and Ram are going to be at the HLS event this afternoon at 4:30 in room 2022. So if you have more questions, please come by and meet other biologists and other people actually working on the genomics field. So please give a warm welcome and a good hand to Ram and Sharon.